Hello students, I am teacher Svetlana, and in this session we will learn about artificial hybridizations and the process of double fertilization, which is covered in the chapter Reproduction in Lower and Higher Plants. Students, artificial hybridization, it is one of the major approaches used in crop improvement. Only the desired pollen grains are hand pollinated and used for fertilization. Now this process is accompanied through emasculation and bagging process. Emasculation, students, is the removal of the anthers well before anthracis, so that you can convert the flower into a female flower. And then bagging is a process where after the process of emasculation is done, or after the desired pollen grains are dusted on the stigma of the receptive flower, then the flower is covered with a polythene bag to prevent any other cross or random pollination, and then it is tagged with the date when the any procedures that were carried on the flower. That process is called as tagging. Students, let us now have a look at the process of double fertilization. Now, what is double fertilization? The fertilization process characteristic of flowering plants in which one sperm cell of the pollen grain fertilizes an egg cell while a second one fuses with the two polar nuclei to produce a triploid body that gives rise to the endosperm. So students, double fertilization is a complex fertilization mechanism in flowering, that is angiospermic plants, and it is considered as a universal occurrence in all angiosperms. Students, the process of double fertilization was discovered by Navastin in liliaceae plants like Lilium and Fritillaria. So students, let us have a look at the process of double fertilization now. After a pollen grain has reached the surface of the stigma, it germinates and forms a pollen tube. We are already familiar with this process when we were studying about the germination of the pollen grain. Now, when it germinates, it penetrates the stigma, style, the ovary chamber, and then enters the ovule. Now, this growth of the pollen tube is guided by the chemicals secreted by the synergids. So, these synergids itself guide the direction of the pollen tube. It usually enters the ovule through the micropylar end. This is termed as sporogamy. Here, this is the structure of the ovary, this is the micropylar end, and this is the chalazial end. Now, when the pollen tube enters to the micropylar end, this phenomenon is called as porogamy. Now, in some cases, it is found to enter through the chalaza, known as chalazogamy. And in some plants, it enters by piercing the intubements, which is called as mesogamy. You can have a look at the image here. This is the chalazial end. If the pollen tube enters from the chalaza and then reaches the egg cell, it is termed as chalazogamy. If it enters through the intubements, that is the protective coating above the ovule or the egg embryo sac, then it is called as the mesogamy when it enters from the intubements. Finally, it penetrates the embryo sac of the ovule through its micropylar end only. Then the next step is, after the pollen tube has entered through the micropylar end, the pollen tube carrying the male gametes penetrates in one of the synergids, and the watery contents of the synergids are absorbed by the pollen tube, which then ruptures and releases the contents, including the two non-motile male gametes. You can have a look at the image here. This is in the first case. You have after the germination of the pollen grain, the pollen tube is traveling through the entire style with the two male gametes. Once it covers the entire distance, it enters through the micropylar end, and the pollen tube comes in contact with one of the synergids, where it absorbs the fluids from the synergids, watery fluids, and it ruptures and releases the male gametes. Now, as the non-motile male gametes are carried through a hollow pollen tube, this process is known as symphinogamy that ensures fertilization to take place. Now, syngamy and triple fusion are the two events of sexual reproduction in angiospermic flowering plants. So here you have the fusion of the two male gametes taking place. In syngamy, the fusion of an haploid male gamete with the haploid female gamete, that is the egg, to produce a diploid zygote takes place. So this happens here where one of the male gamete is fusing with the egg to form the zygote, whereas in the triple fusion, the second haploid male gamete fuses with the diploid secondary nucleus, producing the primary endosperm nucleus. So the second one fuses with the two polar nuclei that are present at the center, and they form the first cell of the primary endosperm nucleus, which is also called as PEM. Students, have a look at this image here. It gives you a clear idea of what happens the moment the pollen tube has reached and entered through the micropylar end. So when it reaches to the micropylar end, it is carrying the two male gametes, and here at the micropylar end you have your two synergids and one of the egg cells. The moment the tube enters through the micropylar cell and comes in contact there, it absorbs the sugary fluids, watery fluids that is secreted by the one of these synergids, and because of which it ruptures. Now the moment it ruptures, it releases the two male gametes into the ovule. Now when it is released there, one of this male gamete will fuse with the egg cell, and the second one will go and fuse with the two polar nuclei, forming the first cell of your primary endosperm. So this is how the process of double fertilization takes place. Students, the primary endosperm develops into a triploid endosperm, and the zygote develops into the embryo. You can have a look at this image here. After the two male gametes are released, what happens next is one of them fuses with the eggs and it forms a diploid zygote, and the second male gamete, it fuses with the two polar nucleus and forms the triple endosperm cell. Now, syngamy is a type of generative fertilization, whereas triple fusion is a type of vegetative fertilization. Now here, both the male gametes participate, and therefore it is described, or it is called as double fertilization. So there is no wastage of the male gametes. Both are utilized in the process of fertilization. Let us have a look at the significance of this process of double fertilization. It is a unique feature of the angiosperms. Now, it ensures that the parent plant invests a seed with a food store, and if the egg is fertilized. Now this happens only if the egg is fertilized. So a seed formation takes place. The diploid zygote develops into an embryo, which consequently develops into a new plant. And then we have the triploid primary endosperm nucleus, which develops into the nutritive endosperm tissues. Now this process restores the diploid condition by the fusion of the haploid male gamete with the haploid female gamete through syngamy. Now it also helps to avoid polyembryos. So these are the important significance of double fertilization. That is, it ensures the development of the zygote, and formation. Further, it gives rise to the formations of seed and fruit. Students, one of the significance of double fertilization is it helps to avoid polyembryos. Now, what is this polyembryo? Polyembryony, it is the production of two or more than two embryos from a single seed or a fertilized egg. So it is termed as polyembryony. Now, in plants, this phenomenon is caused either due to the fertilization of one or more than one embryonic sac, or due to the organization of the embryos outside of the embryo sac. So you can have a look at this image here. You can see here you have the pollen, degenerating pollen tube here. It means that this is the micropilar end, and this is your zygotic pre-embryo. But at the chalazal end also you will see development of an additional pro-embryo. And then we also have something called as adventitious pro-embryos. These embryos develop from the cells of the nucellus. Now, nucellus is the parenchymatous tissue which fills your embryo sac. So, if these cells suddenly start dividing or forms an embryo, these embryos are called as adventitious embryos. This phenomenon is called as polyembryony. Multiple embryos develop. Thank you, students, for participating in this lecture. We will meet soon again with the next topic. Till then, take care, be safe, and keep learning.